Yeah.
Okay, I think that's all the questions.
Yeah.
Thank you again for the time and opportunity to chat about our work.
Yeah, and thank you for a very well laid out pathway going from, your initial questions, how you do validation in several different ways. Really a wonderful seminar. Thank you. So, Siju, now we have a general discussion. Is that right?
Yeah, I mean, we can use the next 20 minutes to, ask some additional questions, and maybe Lloyd and Oliver can give us some directions. And we also have other speakers here as well. If you can turn on your video, Dr. Prentice is here as well. He talked about imaging spectrometry, just in case you guys have more questions. Dr. Denslow is also here. She talked about lipidomics yesterday. So thank you guys. And yeah, you can unmute, or you can put questions in the chat. Maybe I'll just start. I completely understand humans and plants, their ecosystems, but I'm more thinking like, if you want to treat a human disease, for example, you want to do gene therapy, right? So it would be good to identify the genes in human cells and manipulate that. Then you can control the metabolite levels, for example, those associated with breast cancer. So I think this question is more for Oliver. So I think, do you see the importance really identify the metabolites made by human cells? Like you
transformed.
Yeah, we don't even make vitamin C. We don't even make all the amino acid, right?
Yes, no, I mean, there's definitely truth to it, that there is a lot of cellular metabolism that is just building and burning. And that's of course important, right? Building lipids, building complex structures, building proteins, or burning things to get energy. Hugely important. Anything you mess with that, you have huge repercussions, right? Specifically in cancer, of course. There's a lot in that, right? Also dealing with different anoxic and hypoxic environments. However, I just want to make sure that people understand that's not all. 20 years ago, people would have said bile acids are there to emulsify fats in the human GI tract. Today, we understand that a lot of bile acids have additional functions throughout the body by binding to receptors. So people try to say that humans are governed by their brain and plants are not, because they don't have a brain. Well, humans are having different organs too, and these organs have to communicate, just as plant roots have to communicate with plant leaves. And this communication is done by modified peptides, modified metabolites, a lot of interactions going on the chemistry level. So it's not just building and burning, it's a lot of interactions that we need to understand, intracellular, but also extracellular, or between organs. And that is something where metabolomics can help, and where we need better tools to get function.
Yeah, great. I do have another question about these libraries databases. MZ Cloud is so powerful. I mean, they use authentic standard, and those spectra annotated and validated, and they have different levels, MS2, MS3. But if I use, for example, a Bruker instrument, the software cannot really take that library. And the same is true with Bruker libraries. The thermal software cannot take it. So that's kind of, how can we make good use of all the libraries, like kind of make them like a uniform format, so all the instruments vendor software can make good use of it?
Well, I think there's some conversion tools out there, and the vendors are always going to want to proprietarize their library so that they work for their instruments, so they can sell more instruments. But we as scientists have the obligation then to make the data publicly available. And again, there are commercial tools for converting one format to the others. And if not, then there are standard formats, CDF, simple things like that that can be done to allow us to trade those spectral data in a more uniform format. MZ-Mine, other tools now are becoming, or MZ-ML, excuse me, are now becoming available for sharing data.
Yeah, and I would agree. You know, at massbank.us, we also allow people to download in different formats. And if there is a new format coming to the market, we'll be happy to make that available to enable people to download things in the format they need. For MZ Cloud, this is not public, right? This is a private database. So making libraries costs money. And I think that should be also databases cost money, and it should be appreciated by researchers and as well as by agencies that these resources need money. And sometimes, like for MZ Cloud, there's a reason. It's not just marketing, but also to kind of recoup that money to some extent. Even for academics, sometimes they sell their libraries and don't make it freely available. So this is sometimes an issue.
So I would be interested in somebody developing a database or library where you actually had a subscription where your usage was based on your contributions. And I think if we could move towards that model, that it might be a little bit more successful in getting people to share. So that the return on their investment is much more than their individual contributions. But when everybody contributes, then we all benefit to a much higher degree.
Stick and carrot and stick. So you need both in a way. And the most academics want to publish. And so these days, when you publish, you have to upload your data set somewhere. That was not the case 10 years ago. And I hope in 10 years, people will also have to upload spectra. So the databases are there to upload spectra, but people are not forced yet to do it. Well, so it's only when the editor will tell them, fine, we accept your manuscript, but you have to upload your spectra, then people will do it.
Well, I'm all for sharing, but I'm also, I think the database people should be more proactive. I mean, uploading, some of our first uploads to Metabolomics Workbench were, heroic lifts. And, it was multi-weeks and it was very painful. And, that when you put that type of barrier there, you really do discourage submission. And so
I'm totally with you. The problem is, if you just talk to the database guys, then the funders will never know. So all these people should not have a help button, but also I have a trouble button to the funding agency, because I can tell you, because I worked with these people for 10 years, the funding agencies think it's all great and perfect. And of course, the people who organize these databases, including our own, will always say everything is perfect, right? So it's only the users who have to ring the bell and said, well, look, it's not that easy as you make it sound. Also for using tools,
I've tried to do a little bit of it, so.
Yeah, I bet you did.
Well, but you don't want to burn bridges, right?
No,
you
want to be polite and respectful. Yeah.
There's a question in the chat. I think Lloyd, you're probably the best one to answer that. Is essentially to narrowing down and focus on critical metabolites and related genes. Maybe you can answer that one.
Yeah, so it looks like he's interested in sulfur metabolism. I would point you to the Japanese, Kazuki Saito and several of their groups have done sulfur omic type of things, experiments, and published in this area. actually, ultra high resolution instruments can actually, from the isotope fine structure, can tell you and identify specific metabolites in a profile that have sulfur and understanding them. But how could I focus on one of the two most critical metabolites related genes? Well, you have to go through a process of correlation, discovery, correlation, and then prioritization. And the more information and the stronger the evidence are for specific genes, those are the ones that you focus in and start with first. And it's been my experience that it works. Certainly at the most correlated genes, they have proven to be associated with that. So how do you prioritize? Look for the things that have the highest correlation and evidence for relationship to that biochemical pathway, and then start working your way down.
Thank you.
Mhm.
Yeah, this student, I think this person is from Georgia University. It's about 3 a.m. in the morning. So thank you for staying on. Really appreciate
Oh,
it.
God bless people like that.
It's hard work.
Yeah.
Thank
Dedication.
you. Yeah, dedication. Yes. And Dave asked a question, asking from a naive standpoint, has there been much or any work on gas volatile metabolites? How many hormones metabolites might be the field missing?
So we do a non-targeted SPEMI analysis of volatiles. We've done it on mouse urine. We've done it on plant nodules. Apparently, when soybean root nodules senesce, they start emitting volatiles, and the earthworms love it, and they come eat all that bacteria and goo left over from that. So volatiles is a really fun area.
Many terpenes. I mean, just think about how many people work on terpenes and terpenoids.
Yeah,
the whole field of people.
Volatile flavonoids, Natalia, deuterava, and the role of those in plant-insect interaction, multitrophic type of analysis. So there's quite a bit of work out there. And I mean, in our non-targeted approach, we probably see several hundred peaks. A lot of those actually are background from the SPEMI fibers, but you can get a good broad view about those. Great thing about volatile analysis is most of them are small molecules. Thus, it's usually easy to get a fairly confident, accurate mass and molecular formula. Zero chemistry is still an issue, but the chemical space is reduced.
So if I can take this on, even in humans, some people really would like to look at volatiles, for example, acetic acid. And propionic acid is a hot field because that's made by gut microbes and supposed to be healthy, coming from the fibers in the degradation in the gut. And people want to measure it in the blood. It's not easy. I can tell you that. Not easy to find those volatiles because they can easily disappear.
Yeah.
So sampling volatiles is, in a lab setting, not too bad, but if you go in the field or go to humans, not that straightforward. And LCMS has a hard time doing that. So just say.
Yeah, we have more success with LC for short chain fatty acids than GC, but we do many of those that way. And yes, you can't lyophilize your sample, so otherwise it's gone. There was another question about hormones, and it's actually kind of an open end. How many hormones metabolites might be the field be missing? Well, we have a targeted methods for hormones because we know that they're there, but they're very low levels. And so our targeted approach may have 15 or 20 hormones in there. But, any molecule involved in signaling could be considered a hormone, right? So it's kind of how many hormones are we missing? Well, that's a very open-ended question.
Yeah, especially if you consider the intermediates or related hormone-related metabolites or degraded versions. There are a lot of them. People usually don't measure them. For example, we actually measure melatonin, but there are 10 different kind of molecules related to melatonin biosynthesis and degradation. So recently we set up a targeted MRM method to measure all of them. So usually people don't get all of them. So yeah, I think in the chat there's a very interesting question for Dr. Prentice, and he's here. So
yeah, let me look at that. So it'd be amazing to do multi-imaging on live samples, which would have to remain hydrated. Is there any possibility this could be developed, for example, in conjunction with confocal microscopy, which also images via lasers? So there hasn't been a lot of work doing live imaging with MALDI, but there has been quite a bit of work done with DESI, so desorption electrospray ionization, which is another kind of surface ionization technique. And instead of an incident laser, the incident probe is an electrospray probe, basically. And so the electrospray strikes the surface, it kind of entrains material from the surface, and then is sampled into the mass spectrometer. So there's been various iterations of that kind of technology, whether it's an electrospray probe or sometimes a small plasma probe. And that's actually being used in surgical environments right now in a number of research hospitals around the world. So I know at Harvard Medical School, they're doing this. I know at Texas, University of Texas, they're doing this a little bit. And they're using that in the operative environment to monitor tumor margins, basically. So there's a little kind of handheld probe that the surgeon uses and kind of says, this is tumorous, this is cancerous, this is not. So that requires beforehand the analysis of many different samples to kind of build up this database to be able to do machine learning and statistical analysis on all these potential biomarkers and stuff. But it's looking really promising, and it's obviously nice that it's in situ, so you don't have to kind of take the sample, go to the pathologist, wait 5 or 10 minutes. So that's nice. And then additionally, rather than it being, kind of an optical subjective answer, it's an answer based on dozens of potential biomarkers. So that has definitely been done. In terms of the MALDI aspect, I'm not aware of that being done a ton as of yet.
Well, there's atmospheric pressure MALDI capabilities in the market. Has that not been applied?
Not really. It's hard to get something like that approved. If you're looking at like an animal or a human, it's hard to get something like laser burning away tissue approved, via IACUC or whatever.
Yeah.
And additionally, MALDI is all of these are generally surface sampling technology. So it's not like with something like MRI where it's super non-invasive and non-penetrating, basically. So you need the surface exposed, which makes it a little bit more challenging.
Yeah, there's another question for you, I think, from Shruti.
Have you done IMS on plant tissues to get relative quantities and spatial resolution of metabolites? How does this vary from human tissues and animal models? Yeah, we talked a tiny bit about this at the end. Dr. Chen and I have actually been collaborating on Arabidopsis root samples a little bit. And there's a number of other folks that are looking at things like corn leaves and yes, the short answer is yes, plant tissues can be looked at a ton. The things that vary mostly are in the sample preparation aspect. So, rather than sectioning things on a cryostat, things like plant tissues tend to be thin enough that you can actually mount them without sectioning onto a substrate, like a microscope slide. And then depending on what type of tissue it is, is it a leaf, is it a root? That will affect how we kind of have to digest away maybe some of the surface wax layer of the leaf to get at whatever analyte we're talking about. But yes, we can do it. Others have done it as well. There are other imaging technologies in addition to UV MALDI that are being used now for some of this. MALDESI is one of them. So MALDESI, instead of using a UV laser, uses an IR laser. And it's called MALDESI because it's kind of a combination of MALDI and electrospray, but essentially it relies on the native water content in whatever it is you're looking at to do the ablation rather than applying a matrix. So yes, it's definitely being looked at.
So Dr. Fain need to leave very soon. So any last minute question for him? Last second questions. Thank you so much, Oliver. Really appreciate your participation and your talk. Thanks a lot. So in the chat, there is another question about derivatization. So Lloyd, could you comment on that one?
Yeah, I mean, I agree that derivatization is a very powerful tool. Actually, derivatization has been around for decades, if not a century, and it's been a common tool for the analytical chemists to identify specific functional groups, for example. So I believe it is a very powerful tool and broad ranging tool for structural determination. But the question goes on and asks about various derivatization methods in spectral libraries. Unfortunately, the diversity is really not there. Most libraries are based on TMS chemistry. And if you go outside of that space, it's really, it can be crippling because the number of IDs that you can get is not as good. But we've also been working on, in silico derivatization protocols. And I mentioned the tool by Dr. Fong Chu called PlantMet. And in that, the GCMS module, it does allow to do in silico derivatization of all the contents in the database and help you search that space. So with that, it's you can maybe recapture some of the loss that you would lose by doing different derivatization chemistry. And there are chemistries better than TMS, trimethylsilane. And there are others that are more stable, but they've don't have quite the popularity. So it is a challenge, but there are computational tools that now help us address this challenge and complexity. But ultimately, it's still an in silico tool. And I believe that if your biological story is important enough, or if important at all, you need to validate those identifications more empirically.
Thank you, Lloyd. One last question for Dr. Denslow. You mentioned yesterday that you measured the profile about 1,000 lipid species, and you said there is about 2,000 MRM method you're developing. Can you tell us when it will be available? I know your lab also provide services to customers. So when would the 2,000 lipid species method going to be available?
So the 2,000 species is already available through ABSciX. If people want to set that up in their own labs, it's they have the method. But in our lab, it'll probably be later this fall before we have time to get it organized. We have a new person in the lab. He's doing great. He's absolutely great, but it takes a little bit of time to get a new method worked up.
Thank you. Back to you, Sally. You want to give a concluding remark?
Yeah, it's just been a wonderful 2 days. Starting from some both very basic tutorials onto some of the most complex advances in the field from our speakers. I think this has been a wonderful way to reach people around the world and entice them into the field of metabolomics if they're not already there. So I think I should end by thanking Siju for really doing the lion's share of the organization and bringing us all together. So we should also give him a hand, virtual or otherwise. So, and yeah, maybe there'll be a reprise sometime in the future. Given the
Thank you.
success.
Thank you, everyone. Let's thank all the speakers and all the people who have attended the workshop. Thank you. Have a good day, and we will send out the video link, to all the participants.
All right. Thank
Thank
you
you.
for the
Take care.
invitation and getting to chat with all. Take care, and we will
Bye.
see you around.